Yeah.
Okay, so thank you very much. It's a great pleasure to be here. I'd like to thank the organizers and Elizabeth and Lionel and everybody for it has been a very great experience here in France. So what we'll try to do in the next 25 or 30 minutes is to give you an overview about what we are doing using iPS cells to develop new biomimetic models for the human brain. So there are several ways to study the brain. As you know, we can use animal models, you can use post-mortem tissue, you can use neuroimaging, but all of them, they have limitations. So it's interesting, it's important to try to look for a new approach to better understand the brain. One way to do it is to use pluripotent stem cells. These cells are able to give rise to any cell type in the body. So if you give them the correct clues, we can make them behave and become neurons and other cell types in the brain. So this area really changed after the work of Shinya Yamanaka, who is actually a buddy of Kenji in the runner and the marathon. And he won the Nobel Prize in 2012 for describing these types of cells that we call induced pluripotent stem cells. So basically, he got cells from the skin of any volunteer, and then he was able to make the cells behave again as embryonic stem cells. So they are called induced pluripotent stem cells, and with these cells, you can give rise to different cell types, including muscle, neurons, and other cells. What we also do using the same kind of approach is to get, besides using skin, we also are able to get urine. So in our lab, we used urine from patients, and then we are able to make iPS cells from these cells, and then make neurons and other cell types. So one of the main challenges and also opportunities to work with human-induced pluripotent stem cells is to make them differentiate into different cell types. So in the lab, we are creating nerve stem cells, astrocytes, and neurons. And what I'm going to show to you now, this is the summary of my presentation, I'm going to show to you three different cell types plus these brain organoids as new ways to study different aspects of mental disorders, neuronal skin interactions, and the psychedelics and the Zika virus. Seems very different approach, but in all of them, we use the same kind of cells. So we start with neuroprogenitor cells to study mental disorders. So basically, we created these human neuroprogenitor cells from patients with schizophrenia, and then we compare those cells with cells from control patients. I'm just going to summarize this data. Basically, what we found that was most important at that time is that there is a constitutive increase in the level of oxidative stress in nerve stem cells from those patients. It's almost 3 times higher. We published those papers, and we are able to revert it with different compounds. It's interesting because there is a hypothesis that schizophrenia is a syndrome of accelerated aging. And we proposed that this could be a good model to understanding aging associated with oxidative stress in vitro. One other aspect that's observed by the physicians is that there is some anatomical changes in the microvasculature of the patients with schizophrenia. So we decided to check if it was possible to follow angiogenesis and to see if there's any kind of crosstalk between the formation of the brain and the angiogenesis. So we did this model in which we get these nerve stem cells from the patients, and then we apply them to a model of vascularization in vitro. And then we show that actually there is a change. So during the formation of the brain, you can change the formation of the vasculature, and there is a crosstalk, and we also found which would be the proteins involved on that, and this paper is also published. So the other part of my talk, I will present what we are doing with the peripheral sensory neurons to study neuronal skin interactions. So this work is done in collaboration with Lionel Breton and the L'Oreal team, and the idea is to create from the iPS cells human peripheral sensory neurons. We did that. We proposed a protocol that, like, after 2 months, we are able to get these sensory neurons. They have all of the electrophysiological properties, and more interesting, they are able to release substance P, depending on the stimulus that you give to them, including nociceptor modulators and cannabinoid modulators as well. This paper is already available, and now our challenge is try to connect these sensory neurons to the skin. So this is what we are starting to do right now. The idea is to try to make in vitro this crosstalk between skin cells and the human sensory neurons. We just have very preliminary data so far. This is a spheroid of these human sensory neurons in the right side of this image, and now we are going to add keratinocytes to the other side of the plate, and this is what we see. We see the migration of the migration of the process from the neurons that are connected to this ganglia to the other side, where are the keratinocytes. So we still have a lot to do on that, but we are also interested to try to create some kind of human organs on a chips in which we can connect skin, sensory neurons, and also the brain. But it's very tricky to work with the brain in vitro. Of course, there's a lot of layers that are happening during the formation of the brain. So in this scheme, you can see that during the formation of the brain, the cells are migrating back and forth to, too much technology sometimes does not help. Okay. So they're migrating here back and forth, and then they go to the cortical plate. So this kind of formation we miss when you work with 2D cultures, right? So one way to bypass it is to culture those cells as we call human brain organoids. This is the way that we culture in our lab. So they are in these spinning flasks, and they grow as these aggregates of cells that go up to 5 to 7 millimeters. And what we see, we have very cool tools to try to follow them. So we can do tomography on them. We can also follow the formation of these rosettes. And it's a very interesting approach to try to understand what happened outside of these brain organoids. But also, if we basically cut these organoids, we see some of the aspects that are also observed in the fetal brain. So, for example, you have the what we call the ventricular zone here. You have the cells dividing close to this ventricular zone, and then they migrate to form the cortical plate. So several aspects that are observed in vivo, we also can replicate here in this model. What we also can do is to do electrophysiology on these organoids. It's a little tricky, but we can identify local field potential on them. And we are now improving a little bit of the technique, and I'm not sure if you'll be able to see. Here we have one organoid, here's another, and another, and I think you guys can see these dark spots here, right? So we become very interesting to see what would be these dark spots, and then we decided to cut them. And then we realized these are like eye-shaped structures with retina markers. So depending on the way that you culture those cells, we are able to improve the formation of these brain organoids, including some other very interesting parts of the brain as the eye and as the retina. So with that in mind, we decide with this kind of tools, like with the astrocytes, neurons themselves, and also the brain organoids, we decided to explore a little bit more about the use of psychedelics in psychiatry. It's an important issue because we are living in some kind of gap in the discovery of new compounds for example, depression or post-traumatic stress disorder. So it's important to have new compounds. And on the same time, it was common to study psychedelics until the 60s and the 70s. But then because of political reasons worldwide, you see that there is a decrease in the amount of publications that ironically are now starting to increase. And some of these publications are being stimulated by the US Army because of the amount of soldiers that they are returning home with depression and other mental disorders. So we are living some scientists are saying, in this psychedelic renaissance, in which we are trying to understand again how would be the potential of psychiatric potential of the psychedelics. We decided to work on that specifically because in Brazil there is some religions that are originally from the indigenous people that they use some brew of a mixture of plants that has some psychedelic effects. They are used by the indigenous for like almost 5,000 years ago. And then there is a group that we collaborate that are trying, they are using it as a clinical trial. They're using this brew that's called ayahuasca as a way to treat depression. And the results are very impressive. There are several compounds presented in the ayahuasca. And we decided to see, using our models, what would be the cellular and the molecular changes observed in this in our samples with these compounds that are isolated from the ayahuasca. So one of these compounds is called R-mine. So one question that we asked is, what would be the consequence of the exposition of nerve stem cells to R-mine? We found that like R-mine increases the neurogenesis, increases the proliferation of human nerve stem cells. And what's more interesting is that we found that it's dependent on the inhibition of one protein called DYRK1A. What's interesting about this protein, DYRK1A, is that it is a novel risk factor for Alzheimer's. So we are now exploring this possibility, and we are creating brain organoids from Alzheimer's patients, and the idea is to expose them to R-mine. The hypothesis that we're going to reduce the formation of the plaques using the R-mine. This is the work of one master's student in my lab, Karina Carmalina. Okay, the other compound that also happens in a very increased amount in the ayahuasca is called DMT. And then we decided to check what would be the consequences of DMT exposure to brain organoids. It's important to highlight that these brain organoids, they have serotoninergic receptors, and the DMT is able to activate these serotoninergic receptors. We did it, and then we combined a series of techniques, such as shotgun proteomics with different softwares to try to identify the signaling pathways that will be altered after the exposure to DMT. And we found almost 1,000 proteins that they are changed after the exposition to DMT. And there are several pathways. I'm not going into details on any one of them. I'm just going to highlight two of them. So we showed in this paper that like there's an inhibition in inflammation that's basically followed by the signaling of the NF-kappa beta that could be changed after the exposure to DMT. And also we saw increase in the signaling pathway associated with long-term potentiation, memory, and brain plasticity. So this is just to highlight how interesting are these models to try to understand very complex situations, as for example, the use of psychedelics. So this paper was published, and it has a very interesting feedback. Lots of readers, lots of people interested in psychedelics, right? This is my guess. So we conclude that like antidepressant effects of ayahuasca are associated with neurogenesis. This is caused by R-mine, and also with some changes in brain plasticity that would be caused by DMT. We are working more on that, and especially in the connection with the clinicians that they are applying ayahuasca as an official clinical trial in the country. Okay, so just to finish my presentation, I'm going to give you an overview about what we are doing regarding the Zika virus. So, as you probably know, Zika virus is a virus that was isolated in one forest called Zika in Uganda, in Africa, in 1947. And until almost like 3 years ago, nobody really was even studying this virus. It arrived in Brazil probably between 2013 and 2014. One of the hypotheses is that it arrived during the Confederation Cup, that it's the 1 year before the World Cup that also happened in Brazil. And since then, there was a huge increase in the number of children with microcephaly, as probably you saw. So this is the scenario that we had in 2015 in Brazil, almost 200,000 cases of Zika, 11,000 cases of pregnant women, and 3,000 cases of microcephaly. It was very huge when you compare to any other period in the history of Brazil or of the world. And on that time in our institute, we started to receive several cases, and the people from the radiology side started to follow them. And one thing that caught our attention is that one of the most important aspects of the imaging of these children was the formation of these white spots here. These white spots, we call them some kind of calcification. And when you have these calcifications in the developing brain, looks like that you have an interruption of neurogenesis. So we said, let's try our models. Let's try our models to see if the virus was exactly reaching the neural cells. Because on that time, we don't have any clue. We just will have like increasing the number of Zika, the Zika outbreak, followed by the increasing number in the microcephaly. We didn't know if it was the virus affecting the mother, and the mother was, for example, generating antibodies against the child, or if it was something directly into the body of the child, or if it was something directly into the brain of the child. So we did that. We applied the virus to nerve stem cells derived from the iPS cells, and then we did several characterizations on that. We saw that this virus was able to infect the cells and also to multiply inside the nerve stem cells. This is, we show that like it replicates a lot into the nerve stem cells. And when we add it to the brain organoids, we saw a decrease in the growth of them. So there is actually one decrease that goes around 30 to 40%, what's very similar to what we observed in the real child, in the real children, when they're infected in after the third month. Okay, this paper was published. It was an important paper. It actually helped the WHO to conclude that there was really a connection between the Zika virus and the congenital malformations caused by Zika. And then we were in the middle of that kind of challenge of the outbreak, and then we decided to search for drugs that could help us to try to understand a little bit more about the, don't know why it's not following, okay. Then we decided to do a high content, to do a screening about compounds that could be helpful to the pregnant women. So we have a library of compounds that they could be taken by the pregnant women, like, so in the case of an emergency, they could take that. And based on this kind of approach, combined with the iPS cells that we have in the lab, we propose two drugs. One is called chloroquine, that's originally used against malaria, and the other is called sofosbuvir, that's used against hepatitis C. So these are the two drugs that we propose together with other colleagues from Brazil that could be used by the pregnant women in the case of a future outbreak. We were very interested to understand what would be the consequences and how the cells were behaving before they died. Okay, the conclusion of the first paper, okay, the cells are infected and then unfortunately they die. But exactly what happened with them when they are struggling with the virus. So we created an approach in which we infected the cells, but before they have any signs of death or any signs that they're going to go into apoptosis or autophagy or anything like that, we were exploring all of the proteins and all of the gene expression happening there. And based on this kind of combinations of techniques, we are able to identify a network of proteins that were changed after the infection of brain cells with the Zika virus. And we divided these alterations in 4 steps. So first, the cells, they have their machinery being hijacked by this virus for the virus to replicate. And because of that, there is chromosome instability, DNA damage, that would then arrest the cell cycle. And because of that, you stop the formation of for example, the cortical cerebral cortex or the cortical brain. Okay? But one thing that also caught our attention, now I'm going to show to you unpublished data, is that when you look at in the brain organoids, in the sites where the virus were proliferating more, we found something interesting. We found that there is here we go. There is an increase in the infection in the areas in the brain organoids where we have the astrocytes. So this thing caught our attention because the original papers, they were proposing that the main target of the Zika virus was nerve stem cells. And at least looking at the brain organoids, we found that maybe it was not the case. And then we have access to the sections of the brain of the children, of the postmortem tissue, of the babies and fetal tissue. And then we confirmed that actually there is a preference of the Zika virus to infect astrocytes. And it's important because it can explain better what's happening in the brain after they're infected with the Zika virus. We are able, this is another published paper from our group, we are able to isolate from the brain organoids different cell types, for example, astrocytes. In this paper, we compare these astrocytes derived from the brain organoids with real astrocytes derived from the real brain. And they are very similar regarding the protein expression and markers. So we use these astrocytes as a way to understand some of the physiology of the human astrocytes. And we did three different cultures. In one we have nerve stem cells, in the other astrocytes, and the other neurons. And this is what we found. There is actually infection of nerve stem cells with Zika virus, but there is almost 80% of the astrocytes that are infected, and neurons basically not. So this is interesting because it changed a little bit the strategy of how to understand the biology of the virus within the brain. And then we started to work specifically with the astrocytes. We found, for example, that the Zika virus increased oxidative stress in these human astrocytes, and we are able to revert it with ascorbic acid. We also found that there is a change in the stress in the endoplasmatic reticulum of these cells. We also show that like Zika virus is able to damage mitochondria. And this is what we are seeing here in this figure, is that this is in the control situation, 90% of the mitochondria, they have this kind of healthy aspect here. But when they are infected, they change, and almost 70% of the mitochondria, they change in shape, as you can see in this image here. And of course, it's followed by a change in the respiration and in the generation of ATP. And the last part of this, the presentation about Zika, we show that like, besides everything that I just showed to you, there's also DNA damage. And it's very important because when you talk about 3,000 kids with microcephaly, it's clear to see, right? Because it's just you have to measure the size of the head. But probably there are much more children that they were also infected, but they don't have the change in the size of the head. And this data suggests that like you have DNA damage caused in the astrocytes, in the human astrocytes. So maybe you have other consequences that could be not associated specifically with the change in morphology, but that could happen, unfortunately, later in life. That could be, for example, associated with autism or even schizophrenia. So this is the model that we are proposing in this paper. So basically, you have the virus that go into the cell, then it go to endoplasmatic reticulum, where it started to multiply, and then it basically takes the energy from the mitochondria, and because of that, you increase the levels of oxidative stress, and this oxidative stress damage the DNA, with probably very permanent consequences on that. And so this is the conclusion of this part. So human astrocytes are the main target of the Zika virus. The consequences that are caused by oxidative stress and DNA damage probably is beyond what we see in the population of the microcephalic children, and definitely we have to follow a much bigger cohort of children affected by the Zika virus. And there's also one thing that we would like to highlight, we already highlighted to the Minister of Health in Brazil, is that there are some very interesting approaches in which the idea is to use the Zika virus as a way to treat glioma and glioblastoma. It's a very interesting proposal. But what we try to propose here is that this kind of approach should be better discussed, because if the main target of the Zika virus is astrocytes, maybe the consequences of the infection, even therapeutically speaking, should be discussed. So with that, I hope that I convince you that like it's very interesting to work with these brain models, that like there is the technology regarding the use of brain organoids really improved a lot in the last 5 years. There are also sensor neurons in the available, and there is a reconstructed skin. So we still have a lot of challenge, for example, to make the skin to communicate with the sensor neurons, and then to make the sensor neurons communicate with the brain. But it's a very interesting challenge that we hopefully will be able to contribute in the future. With that, I finish my presentation. I'd just like to thank our sponsors, our collaborators in all of the projects that I mentioned to you. This is my team. I'm very proud of them. They are my superheroes in some sense. And this is in the left side, this is my group, and in the middle of the slide, the collaborations that we have, especially in Chile, in Canada, in Argentina, and with L'Oreal. And I really like to invite you to visit Rio and to visit our institute in the future. Thank you very much.
Thank you. Do you have a question for Stevens? Lionel? Tu as un micro derrière toi, Lionel.
So, one more time, I'm so curious of your techniques. So, just to, and we never exchanged on these fields together since a long time, so, but just wondering if there is something that could be linked to skin-brain microbiome axis or gut-brain microbiome axis that could explain, so not Zika pathology, but so as described by many people all over the world in Parkinson, from alpha-synuclein progression from the gut to the brain, and some preliminary hypotheses on Alzheimer explaining that some beta amyloid can come from the skin to the brain in the early stage of the disease. Do you have some discussion or project or something like that on this field, or maybe too early?
I don't know if I understood quite well because the sound goes to your side. So you're asking about if you could play with microbiome in this kind of...
My question is, so do you plan some project on skin-brain axis, skin-brain microbiome axis, or gut-brain microbiome axis, to try to recreate from scratch brain organoid, or using brain organoid to investigate the effect of some commensal or pathogenic strain coming from gut imbalance or skin imbalance in any kind of neurodegenerative disease, for example?
Oh, definitely. This kind of tool could help us to try to create this kind of models. And there are some very interesting recent papers showing some microbiome inside the brain. So one thing that we are not excited in the beginning is that, okay, there's no microbiome inside the brain, and in the kind of model that we have, we should mix them. But our paper, I think that was 2 weeks ago, showed that you have some bacteria working or playing in the brain, in the healthy brain, with, of course, consequence associated with neurodegenerative diseases and other things like that. So we are very open to this kind of approach. We have the tools, and it's just a matter of thinking a little bit harder in this kind of approach to combine microbiome with these techniques that we have there.
In the brain, organoids that you showed with high structure, there is a pigmented part, and I was wondering if you have any details on the type of the cells this is. Is this, for example, cells of the RPE, retinal pigment epithelium, or melanocytes?
Oh, it's a very interesting question. We use a marker for retinal cells. So it's a generic marker for proliferating retinal cells. We didn't use any marker for the pigmented epithelium. We just saw it based on the color, that there are these dark spots rounded, and then we cut. It was, for us at least, looks like at the eye. This is why we use these antibodies against the retina. But this is the only thing that
shape, perhaps, of the cells? Is it a spindle shape, morphology, or something more epithelial? Or?
In the eye-shaped area, you mean? No, we didn't. We just see like this. It looks like a very primitive kind of retina, right? It's like I used to work with the mouse retina 25 years ago, and it looks like you have the there's no really clear plexiform layer or the retinal ganglion cells. Basically, it's just a shape of cells similarly, but they are rounded like that in some kind of a ball. This is what we have so far. We are able to keep these organoids up to 10 months. So maybe you have to push them to go a little longer. I have a former student of mine now in Harvard, work with Paola Lotta. They are able to keep these organoids for 3 years. So maybe after longer times, it will be easy to identify and even to expose them. Actually, one of the former postdocs in the Lotta's lab, she was able to expose some of these organoids to light, and they respond. So maybe there is, depending on the protocol, you have to improve it a little bit to get better retinal cells.
Okay. Yeah, here on the back, just in front of you. Yeah. Regarding the Zika part, actually I was wondering, you are proposing that astrocyte is also a target for Zika. Yeah. I was just wondering, or can you comment just on that? Is it possible that during, for instance, brain-human development, one of the first targets would be the neural stem cells? That would possibly explain why the brain is smaller and developing, and that maybe after that, after development, one target would be astrocyte, actually. So depending on where we are, at what time you can change the target. And just more specific question, when you were showing that mitochondria were damaged, I was wondering whether you increase mitophagy or whether mitophagy is impaired.
Okay, I will start with the second question. We don't know. We didn't follow if there's more mitophagy on that. We saw that there's more autophagy in general, but specifically regarding the mitochondria, we didn't follow to see if there is more. It's a very interesting one. Regarding your first question, we didn't have time to show it in our first paper. Like if you get very primitive organoids, or as we call neurospheres, there is a complete destruction of the tissue. If you get a little bit after 2 months, they started to, there is a reduction in the growth of them. Comparing with what we see in vivo in this post-mortem tissue, we of course have a lot of cell death in the ventricular zone, but more than that, you have the formation of the cortical plate is completely modified. So you have much more of the gyra and sulci that's completely different from the normal one. So one of the proposals, at least based on what we see in vivo in the patients, is that not only you are killing the nerve stem cells, the pool of that, but you are killing the radioglial cells that will give the clues or the pathway to the nerve stem cells to migrate to the cortical plate. So sometimes, like the nerves or even the young nerves, they are not dying, but they are making a mistake to where to stop. This is why we believe that, like, okay, nerve stem cells, it's a target, but the main target is astrocytes, because it's not only explain the death that's happening in the ventricular zone, but also the change in the position of the cells that they are migrating, but basically they are lost because the astrocytes, they are infected.
Would you consider that Zika virus could induce senescence in astrocytes? Because all you described correspond to senescence, NF-kappa-B, mitochondrial alteration, ROS, DNA damage. And if yes, did you look whether there is a chronic inflammation in the brain of the pathology?
It's a very interesting point. Definitely there is inflammation there. There's a lot of microglia also that are being recruited after the infection. But at least in the case of astrocytes, after a few days they die. So maybe if you get like more older astrocytes, they are not going to die, they're just going to senesce. But at least with the ones that we're seeing here, there's a lot of reactive gliosis in vivo in the patients, and in vitro in our models, they also have all of the phenotype of inflammation or reactive gliosis, but after a period, they basically die.
Professor Yamashima.
So yeah, it has been reported that astrocytes is involved in the development of chronic itch at the spinal cord level. So do you think astrocyte in the spine is also a target of Zika virus?
That's a very interesting point. Like we know about the Guillain-Barré, right? That's also associated with the infection in adults. So this could be a target. When you look when we look at basically in all of these sections of the postmortem tissue from the babies, and we saw, depending on the stage when the infection happens, even a change in the formation of the spinal cord that could be followed by reactive gliosis. So definitely there is an infection of astrocytes in the spinal cord, but the consequence, it's some kind of a speculation, but based on some of the patients that we also saw there in our institute, you have Guillain-Barré associated that could be also be linked in some sense to the astrocytes in the spinal cord.
I see. So there is no good report about allodynia or chronic itch might be involved, might be regulated after Zika virus infection.
Yeah, not that of course, like one of the, if the person has the believes that she has Zika virus, she goes to a health place, and then she start to complain about rash and about itch. So definitely it's a way to confirm the symptoms. But I didn't know papers showing, like as we are seeing in the other presentations, how would be the mechanism that associating the infection with itch. But definitely one of the consequences, the rash and the itch.
Okay. Thank you.
Thank you. Other questions? No? So, thank you. Thank you so much.